Our last speaker is Rebecca Saxe. Rebecca is a neuroscientist. She works on social cognitive neuroscience. She has a lab at MIT. It's called the Saxe Lab. And she focuses on the brain, how it constructs abstract thoughts, what happens in our brain when we try to consider other people's thoughts and other people's state of mind. It's really fascinating when you think about it, because we're not only talking about what happens in our own heads, but how that kind of meshes with what's happening in other people's heads. Her research has implications, she believes, for things like autism, developing artificial intelligence, even mediating international conflicts, so enormous practical application. And I want to read you a quote from Rebecca that I really like. She's talking about the brain. She says, "The machine that we use for thinking about other minds, our brain, is made up of pieces that we share with all other animals, with monkeys, mice, and sea slugs. But when you put them together in a particular way, what you get is the capacity to write Romeo and Juliet." So I really like that. So Rebecca Saxe is our next and final speaker.
Thanks, Sasha. Thanks. Well, so this has been a long and varied and kind of amazing day, and I think it would help to get back into things, get everybody awake again, to start this talk with a game in the spirit of the talk that we just heard, and in particular with an experiment. I'm a neuroscientist and a psychologist, and I do experiments on people, and so I'm going to do an experiment on you. In this experiment, I'm going to tell you a story about a person. Her name is Grace, and I'm going to ask you to make a moral judgment of her action. So at the end of the story, I want you to tell me how much blame Grace deserves for what she did in this story. The more blame, the higher your hand, okay? So if she deserves lots of blame, raise your hand high. If she deserves only a little blame, raise your hand low, and everybody has to play, okay? All right, so in this story, Grace is on a tour of a chemical factory, and during a break in the tour, she goes to get coffee, and another girl on the tour asks for a cup of coffee as well with sugar in it. So Grace goes to the coffee machine, and next to the coffee, she sees a jar of white powder labeled sugar. And so she puts some of the white powder in the coffee and serves it to her friend. Unfortunately, that white powder was contaminated by a dangerous toxic poison, and so when Grace puts it in her coffee and her friend drinks it, the friend dies. How much moral blame does Grace deserve for putting the powder in the coffee? Now, what if the story was slightly different? What if the jar of white powder was labeled dangerous toxic poison? So when Grace saw the powder, she thought that the powder was dangerous toxic poison, and she put the powder in her friend's coffee, and her friend drank the coffee and died. Now, how much blame does Grace deserve for putting the powder in the coffee? Good. Okay, so what this shows is a remarkable and very general feature of human moral judgment, which is that when we evaluate other people's actions, we're not only evaluating what happened. We're not just evaluating if they did good or bad, if they caused harm or good. We're evaluating their intentions, what was in their mind when they went ahead and did that action. And your judgments here that make a big distinction between whether she thought she was going to kill the other girl or not, look like the judgments of a population we call typical human adults. They're MIT undergraduates. So these guys also make a big distinction between what the person did and what she thought she was going to do. And this ability to take other people's intentions and beliefs into consideration is totally ubiquitous in human cognition. We're using it all the time. And underlies both ability to do simple tasks like perceive movements as the movements of an actor or intentional agent, all the way up to the ability to understand really complicated and arcane sentences like this one apocryphally attributed to Alan Greenspan, who apparently said, "I think you understand what you thought I said, but I'm not sure you realized that what you heard was not what I meant." Normally I can do that. Okay, so this ability to keep track of other people's thoughts, to understand what they're trying to tell us about their intentions, is a deep part of the human mind and cognition. It goes beyond the general abilities we have as people to solve complicated problems like calculus and design bridges, and especially necessary when we're trying to get people together and deal with one another, when we're evaluating their actions, when we're predicting their actions, when we're strategizing with or against each other, to flirt, and perhaps most importantly, to teach, right? Perhaps the most astonishing social action that human beings engage in is the thing we're doing right now, where we all get together to basically for free donate knowledge to the children of strangers. No other species does that, right? So this human social cognition, that deep intuition to give ideas, thoughts, to interpret what's inside other minds, It turns out, and this is the topic of my research program, depends not only on our general capacities, the general abilities of our human brain, but on the evolution of a particular mechanism or brain region that is specifically for thinking about other people's thoughts. And I'll start by telling you a little bit about what that means, and then a little bit about what we can do once we know that. So research in my lab mostly uses an fMRI machine. So we have people come into our lab, they lie down on their back, we slide them into a dark tube, and then we ask them to engage in social cognition. And you might wonder, how do we get people engaged in thinking about one another if they're lying by themselves on their back in a dark and noisy tube? And the answer is the way that we often learn about each other, which is by asking people to hear each other's stories. We tell people short stories about somebody's experience and ask them to think about those stories and get engaged in the person's thoughts the way we've been doing all day long and listening to people's talks. So you might read a short story about Maggie, who's in love with a man who sends her a text message he meant to send to somebody else saying he doesn't want to see her again. Hopefully that invokes in you a rich representation of how what Maggie is thinking and feeling. And we contrast that with other stories that tell you about other things about people. For example, a story about a physical sensation, like cutting yourself on a knife while doing the dishes. Each of these kinds of stories gets you thinking about a person, but in a different way, right? One is more about their minds, the other is more about their bodies. And this experiment goes on. You're reading, each story tells you a little bit about a character and their experience. What we're looking for is regions in the brain where the amount of activity or the amount of oxygen going to that brain region, as it would to a muscle, goes up when the story is about somebody's mind, when what you're doing is thinking about somebody's thoughts. And so we look across the whole brain at the blood oxygen signal, and we're looking for places like this where the amount of activity in that brain region is going up just when the stories are making you think about what's inside the characters' minds. Now, over 10 years and hundreds of participants in these experiments, what we've now learned is that stories that make you think about somebody's thoughts, compared to stories about pretty much anything else, get increased activity in a group of brain regions, sometimes called the social brain or the theory of mind network, that basically get more active the more you're thinking about somebody else. And these brain regions are incredibly robust and specific. We can find them in basically each individual person that comes into the lab. This is just 3 people out of the hundreds of people we've scanned. Not just typical human adults or MIT undergraduates, but people of all demographics and age and gender, all show activity, different across people, but in approximately the same region and in the same amount, just when they're thinking about other people's thoughts. So what does having a brain region like this let you do? Well, one thing I told you before is that one place in our lives when we really use consideration of other people's thoughts and intentions is when we're making moral judgments of their actions. And so one thing we showed, for example, is that if you look at the pattern of activity in an individual person's brain, in this brain region that I'm talking about here, this is the right temporoparietal junction, or right TPJ. If you look at the pattern of activity in that person's brain region while they're reading a story, what you can do is predict the moral judgment that they'll make of accidents versus intentions to that story. So in this plot, each dot is a person. And what I'm showing you on the x-axis, so along the length, is how different were the patterns of activity when they were thinking about intentionals versus accidental harms. And going up and down is, for that person, how much worse did they say in their moral judgment that it was to harm somebody intentionally versus accidentally. So we know that information is in there. Another thing we can do is actually interfere with function in that brain region and show that we can change people's moral judgments. So this is a tool called TMS, so transcranial magnetic stimulation. It's a magnetic pulse, which I'm showing you by showing what happens when you put a quarter on it. And now I'm going to put it on my own brain over the part of the brain that controls my hand. And what you'll see is that when the pulse goes off, it makes my hand do just a small movement. Basically, once the pulse gets all the way through the skull to my brain, creates a small burst of disorganized firing in the brain region that makes a small pulse in my hand. So we can ask, what happens when we apply the same mechanism, the same pulsive magnetic field, but now to this critical brain region, the right TPJ, when we're asking you to make moral judgments, compared to when you are getting TMS, but to a different brain region. Okay, so this is when you're getting it to a different brain region, and this is just a measure of how much do you use beliefs in your moral judgment? How much difference does it make what she thought she was doing? Now we move the pulse to the right TPJ, and what you see is that basically what she thought she was doing matters less. We shift moral judgments from depending on what she thought she was doing to what really happened. So that's one thing you can do with this brain region. But it goes beyond just whether you intended to cause harm or didn't. It also depends why you thought what you thought, right? So we can look at just stories where Grace says she didn't intend to cause harm. In all cases, she thought the powder was sugar. But we look at why does she think the powder is sugar. This is really important in actually evaluating other people's actions. So I told you a story where she thought the powder was sugar for a pretty good reason. It's in a jar of white powder next to a coffee machine labeled sugar. That's a pretty good reason. But if I change it to she just guessed, right? It's an unlabeled jar of powder in a chemical factory. Well, now actually it's not so okay that she put it in the coffee, even though she didn't intend to cause harm. And adults' moral judgments appropriately reflect that. We say if she had no reason to think it was sugar, she deserves a lot more blame for putting it in the coffee. And again, we can look inside this brain region, the right TPJ, at the pattern of activity, and again see this difference. We can tell whether you're reading about somebody with a good reason or a bad reason. So that's true in adults. But one thing we wanted to know is what about in children? These are really complicated thoughts, and it actually takes kids a really long time through most of their school years to learn how to sort out these differences, what you intended versus what you did, why you had those beliefs that you had. So we wanted to do a version of this experiment in kids. We didn't at first want to expose kids to dozens of stories of people committing murder. And so we wrote new stories, not about morality, but that just have people coming to conclusions for good or bad reasons. So for example, this is a story who comes to the conclusion she's finally going to get a puppy. And we change what her reason is. Either she has a pretty good reason, she sees a newly installed doggy toy in the kitchen, or she has a pretty bad reason. She's just guessing based on a few toys that are lying on a cluttered kitchen floor. We bring kids in for this experiment. This is what kids look like while they're getting ready to be run in our experiments. This is actually a model of the fMRI machine built for us by a 3-year-old participant. It's remarkably accurate, better than I could do. OK, so then we want to know, in kids, how does the emergence of this neural signal relate to their acquisition and learning of these concepts? So the first thing to know is there is important learning going on. So if I just ask you, how sure are you that Julie is going to get a puppy based on the story that I've told you? Adults say, well, it really depends on the evidence. If there's a newly installed doggy door, I'm pretty sure she's going to get a puppy. But if there's just toys on a cluttered floor, then who knows whether she's going to get a puppy or not? Kids, by contrast, are less sensitive to this difference and basically say if she thinks she's going to get a puppy, she's probably going to get a puppy, right? OK, but that changes with age. So this is over a group of 30 kids aged 7 to 12. This is how confident they are that Grace is going to get Julie's going to get that puppy if she had the bad evidence. So this is in the cluttered kitchen case. And what you can see is that while the 7-year-olds are pretty sure she's getting the puppy, by the time they're 12, they're more savvy and skeptical. They think she may think she's going to get the puppy, but I'm not that sure. So over this age range, we've got developmental changes. Kids are coming to evaluate not only what do other people think, but why do they think it? What were their reasons? And corresponding with that, if we look in these kids in the same brain region over time, what we see is that as kids' right TPJ starts to separate, the patterns of activity start to separate stories with good evidence versus bad evidence, so the kids themselves are beginning to separate whether she has good evidence or bad evidence. So the emergence in the brain of this distinction is correlated with the emergence in explicit behavior and evaluation of the same distinction. So that's the kind of thing I mean by saying we have a brain region for thinking about thoughts. We have a brain region where the amount of activity, how much it gets going, depends on whether you're thinking about somebody else's thoughts, and where the pattern of activity, what the different neurons inside it are encoding, reflects distinctions that matter about other people's minds. Did they think they would cause harm or not? Did they have a good reason or not? So now that we know that, can we use that to do anything else? And I'm just going to show you 3 things, 1 that we've done and 2 that we're doing, to try to use this idea to get purchase on other bigger questions. The first reason why I like working on this kind of topic is because it lets us go back to old philosophical puzzles that we might all have wondered about in college with a new empirical eye. And one of these questions is, how stuck are we in our own lives? Or on the other, more optimistic end, how much capacity do we have to understand the minds of people who are really different from ourselves? Now, neuroscientists have tended to tell us we're very stuck in our own experience, that our ability to understand other people depends on being similar to other people. The more similar you are to somebody, the more you can understand their mind. The more different you are from somebody, the less you could ever understand what they've experienced. And that, to me, seems deeply pessimistic. I think if you have an amazing storyteller, like some of the talks we heard earlier today, a storyteller can get you into the mind of almost anybody. Right? Can teach you to think about the experiences of people across distant worlds and cultures and backgrounds. But how would you take this question? So here's a version of this puzzle. This is a 2,000-year-old version of this puzzle from China. Shuangzi observed, see how the small fish are darting about? That is the happiness of the fish. You are not a fish, said Huizhe. How can you know the happiness of the fish? And you not being I, retorted Shuangzi, how can you know that I do not know? So to think about this question empirically, what can we know about another person without being them or being similar to them? I wanted to take a case where we could make it concrete. What aspects of the other person's experience have you had or not had? And so I translated this into a specific version of the question, which is, what can congenitally blind people learn about sight? So there's a group of people who've never had a class of mental experiences they've never seen. But they live in the world of the sighted. The people around them talk about vision and act on their vision all the time. So this seems like a test case to ask, what is it possible to learn about an experience you've never had? Now, there's already hints that blind people can learn a lot about sight. Some of this comes from blind painters. So these are paintings by Eşref Ermcan, a Turkish painter who's blind. And you can see that he knows an amazing amount about vision. He knows about occlusion. He knows about perspective. He knows about the colors of shadows. So this shows that it is at least possible for somebody who's never experienced vision to learn a tremendous amount about what vision is like. But he has put a tremendous amount of effort and expertise into this. And anybody in the room who's an expert at anything, you know that expert knowledge is a little different from normal knowledge, right? When you put out your whole life into learning a domain, you have a different kind of knowledge. And so we wanted to know, what about just regular people growing up blind, but in the world of the sighted, what do they come to know about the experience of vision? And we wanted to get beyond just what they could tell us verbally, because some people have been suspicious that blind people learn to talk the talk. They say words about vision. They can parrot back to us what we've told them, but they don't really understand. And so this is where being a neuroscientist gives us an opportunity to sort of look under the hood and say, what we mean by really understand is have the same representations in your brain when you're thinking about that kind of thing. And so we had both sighted and blind people come into the lab and hear stories about people having experiences based either on vision or audition. So for example, you might hear about Abigail, who while she's cleaning her room, sees an old love letter and recognizes the handwriting as that of her boyfriend. So that's a very visual experience. Or you might hear the story about Abigail cleaning her boyfriend, who hears footsteps coming down the hallway, and it sounds like her boyfriend. So the footsteps is an experience the blind people have all the time. In fact, they're better at it than sighted people are. The handwriting is an experience they haven't had. There's nothing like handwriting in the auditory domain. In sighted people, we showed that this same brain region that tracks what you believe and why you believe it also tracks whether you got your information from vision or audition. And then in the blind participants we had come into the lab, we found exactly the same pattern. So again, in the same brain regions, they're tracking whether other people got their information from vision or from audition. So that's a little bit about what we know so far from these brain regions. We know that we as human beings can learn about other people's thoughts and experiences, even when they're different from our own, and that our ability to think about thoughts reflects functions not just of our amazingly capacious human brain that lets us solve all kinds of problems like algebra and calculus and building bridges, but is specifically designed for thinking about other people's thoughts, and that this is true both in adults and in developmental change in school-age children. So now I'm going to tell you a little bit about some of the things we're not done with, new projects that we're just working on now that are, I think for most scientists, always the thing you haven't finished is the thing you're most excited about. So what's next? One thing I'm really interested in is learning more about development. I want to know both how can we promote development of these brain regions, so we show there's developmental change in kids. What can we do for kids to help these brain regions develop more and better? And perhaps more fundamentally, how do we come to have specialized brain regions for this at all? Imagine a human baby that starts with nothing, right? How does their brain develop or acquire regions that have specialized functions? And we know almost nothing about the first years of development of human brains, mostly because it's really hard to study babies' brains. We know, though, that babies' brains look really different from grown-ups. So this is a grown-up brain. And this is a baby's brain, a 4-month-old brain. And the reason the baby's brain is so much darker is because the connections between brain regions haven't grown yet. Basically, the white stuff in the bigger image is all the wires connecting different parts of the brain. And in the baby's brain, those wires aren't there yet. That's in a 4-month-old. That's actually my son when he was 4 months old. And one of the things I want to know is, so you start there with all the bits of your brain unconnected. How do you acquire the specialized functions that you will have by the time you're 4? This is him helping out with the experiment. So that's one thing that we're working on. The other thing, which Sasha mentioned, that I'm interested in is how to think about how adults use these brain regions, not just in laboratory contexts, not for simple judgments, but for the hard problems. And I think the hardest problem is for thinking about the beliefs and motivations of the people we most deeply disagree with. When we're deep in conflict with somebody, it's very tempting to lose all empathy, to decide that they are ideological and dogmatic and that nothing they say is based on reason. And so one of the things we've been studying in the lab is the brain mechanisms that underlie this tendency to believe not just that the people you disagree with are wrong, but that they're crazy, right? That it's fundamentally irrational to hold that view. And we've been doing this by showing people arguments from the other side of a conflict and saying, not do you agree with this, but could somebody reasonably believe this? Of course, we get very strong answers of no, they couldn't. And we're looking at what brain regions are related to that judgment that you couldn't even reasonably believe this. We've looked at this across fairly intractable conflicts, including Israelis and Arabs who we scanned in 2009. And then we replicated the same pattern in Democrats and Republicans right before the last election. So this suggests that there's a challenge here, which is how to make people recognize not that other people necessarily are right, but that they might have good reasons for their arguments. And we've been interested in developing this not just as a basic science question, but also to know how can we help programs, in fact, like the programs we just heard about, do a better job of making people hear the perspectives of those who come from other places or other views. And we've done this in part in collaboration with a group called Celia that runs virtual contact programs. These are programs where American university students, as part of their coursework, do collaborative coursework with university students in similar classes in the Arab world and in Arab and Muslim cultures. So throughout the entire semester, they're doing work together through a virtual connection with peers, but in the Arab and Muslim world. And we're interested in the capacity of these kinds of collaborative efforts to change people's views of the reasons that other people have and of their minds. So our basic design is that we have added survey measures for the moment. We dream of neuroimaging, but that's too hard for now. For now, it's just survey and behavioral measures before and after this virtual contact. And the last time that we did this, when we had both American students and control participants, so these are students at the same universities attending similar classes but not involved in this program, and we evaluated their attitudes towards Arabs and Muslims before and after the semester that the program ran. But it provided a particular opportunity to test the power of these programs because we did this in the spring of 2013. And so right before we tested people's attitudes towards Arabs and Muslims, there was the Boston bombing, which was widely advertised as an act by initially Arabs and then Muslims aimed at Americans as part of a cultural conflict. And so we could look at how having participated in a semester-long virtual exchange program changed your attitudes towards Arabs and Muslims in the face of this very strong threat. The first thing that we found is that in the control participants, so the people who hadn't participated in this program, at the end of the semester compared to the beginning of the semester, they felt less warmth towards Arabs and Muslims, and were more convinced that there's a fundamental clash of cultures between the Western world and the Arab and Muslim world, perhaps unsurprisingly. What we found in the kids who had been in this program was the opposite. Even immediately after the Boston bombings, these kids felt more warmth towards Arab and Muslim peers. And we're less convinced that there was a fundamental clash of cultures between these two groups, suggesting that having been deeply involved in hearing and evaluating the other's perspective was in some way insulating against the local threat of this specific event. So that's some of our research, just to give you a sense of the bigger picture. I think that what this research offers us as an opportunity is to use neuroscientific and scientific methods to approach what I think are very fundamental big questions about how we engage with one another, to teach us how to grow and use better the brain regions that we evolved for thinking about other people's thoughts. Although I want to finish with the famous line from Philip Roth, just to temper our enthusiasm, who said that getting people right is not what living is all about anyway. It's getting people wrong that is living, getting them wrong, and on careful reconsideration, getting them wrong again. Thank you.
Thanks, Rebecca. I have a question for you that's very similar to the one I asked Nathan Nunn, who does the sort of economist historian work, which is in this vast world of neuroscience, when there's so many pieces you could have honed in on. What drew you to this one? What made you fascinated with this, and this is what you wanted to focus on?
I think in any scientific personal story, it's always a combination of luck and happenstance and some personal investment and help and mentorship, all the things we've heard about today. So in my case, I was drawn to working on social cognition as a problem in neuroscience because I wanted to work on a problem I could explain at a dinner table. I had previously worked on a very subtle detail about attention, how we choose which features to attend to, and I decided it would be more fun to work on problems that seemed interesting to other people. Also, I think, honestly, our capacity for social cognition is one of the most remarkable things about the human brain. It's one of the things we do most obviously differently from any other animal. And I study humans. I think it makes sense when you study humans to study the things that humans are best at. So that's the second reason. And then luck. It turns out that there are brain regions, specific brain regions for thinking about other people's thoughts that are really easy to find. And there's lots of things we do with our brain that aren't easy to study because it's just really hard to find the parts of the brain that are responsible with the tools that we have.
What are examples of that? The hard ones, the hard to find ones.
Oh gosh, like love. That's really important, but really hard to study as a neuroscientist. Actually, I think the hardest class of things to study, and perhaps also the most important class of experiences we have, is anything you can't recreate, right? So the whole scientific method depends on studying something repeatable, right? You have to create it and then create it again and then create it again in order to study the conditions that underlie it. Basically, with some very few exceptions. And so anything that happens once to one human being and never again is kind of out of our grasp.
I found myself wondering as you were talking whether what you have learned about neuroscience and sort of the mechanics of the brain in your work, do you think it makes you think or act or behave differently? Since you're constantly conscious of how our brain works, do you think it affects you?
I will say this. I had a baby. And to me, the most obvious thing you would do when you have a baby is watch his brain develop. And I think not everybody reacts that way.
And did you end up thinking you could have a more active role in it because of so much you know about the brain? You could.
No, but it was so cool. We scanned him at 1 month, 3 months, 6 months, and 8 months.
Was that your baby's brain we saw up there?
Yeah, that's my baby, yeah.
You know, last year, President...
I've got another participant on the way, actually.
The Obama administration last year announced its BRAIN initiative, which is based on the Human Genome Project. It's trying to map the activity of every neuron in the human brain. I'm wondering how excited you are about that, and if they're hoping, is there anything you hope in particular they discover in the process?
So actually, they don't pretend they're going to map every connection or every neuron in the human brain. That's clearly not going to happen, nor is it actually important. It's sort of the wrong level of analysis. It's like saying that in order to understand a person, you want to know where every one of their atoms are, right? You actually want to know more like where their organs are, not where their atoms are. And similarly in the brain, we want to know the kinds of processes that occur, like what are those groups of neurons doing as whole groups. Each individual neuron is probably too much detail. And unfortunately, that's not exactly what they're going for. But it's true that right now is actually a tremendously exciting time in neuroscience. neuroscience, mainly because of a huge range of new tools that have been developed over the last 10 or 15 years that make things possible that have been never possible before. Probably the most exciting class of which is a set of things called optogenetics that let you label. This is a tool that lets you pick a specific neuron or group of neurons or region of neurons based on any criterion you want. You can find them, you can watch them do their work basically by making them light up whenever they're firing, or you can actually make them fire by shining light on them. So you can pick a group of neurons in a brain region and control at a millisecond time scale whether they're firing or not. And in the first demonstrations, people have already shown you can use this to drive a mouse. You can control whether it's going left or right by firing different parts of the navigational system. You can make it scared or not scared of a new environment by firing neurons in the parts of the brain that control mouse's anxiety. So this is completely revolutionizing non-human animal neuroscience. This is not permitted in humans. We never do this to people. And so many of the most exciting technological advances that are going on in neuroscience are going on in non-human animals. And will yield incredibly important discoveries for both understanding how our brains work and also for treatment of neurological diseases, which is a huge topic right now. Basically, as we have solved other problems, as we no longer die of car accidents and smoking and cancer and heart attacks, more and more people are going to get old enough to get Alzheimer's and Parkinson's and other brain diseases. And for any of us who have had family members, as I have, who experience Alzheimer's or Parkinson's or stroke, you know that brain diseases in some ways are more devastating than any other way to die slowly, because you lose yourself before you lose your body. And so the push right now to understand how brains work so that we can treat them more effectively, I think, is an incredible moral motivation. And I hope we get there before I'm old.
Rebecca Saxe, thank you. Good to meet you.